So welcome to my YouTube channel. Today we are going to be talking about glycosylation and the different types of glycosylation. Alright, so first, in glycosylation or glycosylation, anyhow you pronounce it, in glycosylation, we are actually producing carbohydrates by modification of proteins or lipids. So we modify proteins to give us carbohydrates. We modify lipids to give us carbohydrate. That process of attaching sugar to either amino acid proteins or lipids to give us carbohydrate conjugates is called glycosylation. So glycosylation, just know that we are attaching carbohydrate to something, or we are attaching something to carbohydrate to form carbohydrate. Now, in glycosylation, we have two major types of glycosylation, and the two major types of glycosylation are N-glycosylation and O-glycosylation. So I'll be taking them one after the other, the N-glycosylation and the O-glycosylation. Now, let's start with the first one, the N-glycosylation. So this N-glycosylation is actually from N-glycosidic linkage. So here we have our N-glycosidic linkage. And please take note, it is important to note that N-glycosidic linkage is formed between oligosaccharide and the nitrogen, the amide nitrogen of asparagine. I'll take that again. N-glycosidic linkage is formed between oligosaccharide and the amide nitrogen of asparagine. The amide nitrogen of asparagine. It must be asparagine, not any other amino acid. Not any other amino acid. Now, that's for N-glycosidic linkage. Now, if you look at this, if you look at the structure of asparagine, now you see that asparagine is, now look at this structure on the board here. I'm drawing the structure of asparagine. Okay, so this is the structure of asparagine. Now you can see that this is an amide functional group. This one here is an amide functional group. So here, if we link an oligosaccharide, that is few sugar units, if we link few sugar units to this and this amide nitrogen of asparagine, what we are forming is N-glycosidic linkage. So what we are forming here, this linkage here is N-glycosidic linkage. So in N-glycosylation, we always have oligosaccharide plus, so linked to asparagine, X, and serine or threonine. Let me explain. This is the oligosaccharide linked to asparagine. This X here can represent any amino acid except proline. So you know proline is an amino acid. So this X here can represent any amino acid except proline. Then we have serine or threonine. So if you have something like this, this will give us our N-glycosylation. So in N-glycosylation, oligosaccharide is linked to the amide nitrogen of asparagine. Sorry, asparagine is supposed to be ASN. Asparagine. Then from asparagine, we have an amino acid, any amino acid except proline. Then we have serine or threonine. Alright, so that is N-glycosylation. And N-glycosylation is used in protein folding. It is used in protein folding. That's one function of N-glycosylation. So it is used in protein folding. It is involved in protein stability. That's in the stability of protein. So when the protein attaches to oligosaccharide, it makes it stable. So another function of N-glycosylation is it is involved in protein sorting and protein degradation. So it's involved in protein sorting and protein degradation. Alright, now this N-glycosylation takes place in the endoplasmic reticulum. So it starts in the endoplasmic reticulum and it continues in the Golgi apparatus. Please take note, that's very, very important, that the synthesis or the N-glycosylation process begins in the endoplasmic reticulum and continues in the Golgi apparatus. All right, so that's that about the N-glycosylation. We're going to look at O-glycosylation now. All right, so I'll be looking at O-glycosylation, but before then, let me emphasize on something. In N-glycosylation, we said the function is, I've given you some of the functions, stability and the rest, but one major thing I want to emphasize is that stability, it prevents protein degradation. Remember I told you it's involved in protection against protein degradation, it's involved in protein sorting, and it's involved in, I said that. Now, that protein degradation is what we term proteolysis. Proteolysis, lysis of protein, breakdown of protein. So it is N-glycosylation that is involved in doing that. So I wanted to just spell out that. Now, O-glycosylation now, in O-glycosylation takes place majorly, primarily in the Golgi apparatus. Remember that N-glycosylation starts in the endoplasmic reticulum and continues in the Golgi apparatus or Golgi body. But now, O-glycosylation is exclusively taking place in the Golgi apparatus. Now, in O-glycosylation, sugar units are added to the oxygen on the hydroxy group of either serine or threonine. Now, you know serine and threonine, they are OH containing amino acids. They are OH containing amino acids. Now, this serine here, if you look at the structure of serine, let me use any of them. Let me use serine. Now, this is the structure of serine. If we attach a sugar unit to this oxygen from the hydroxy group of serine here, if I attach a sugar unit here, what I now have is O-glycosylation. Remember, in N-glycosylation, we're attaching it to asparagine, the N-amide nitrogen of asparagine, but in O-glycosylation, I'm attaching the sugar on the oxygen of serine or threonine. And one thing you should note here is that here is not the oligosaccharide we are adding. We are attaching the sugar one after the other, stepwise addition of sugars. But in N-glycosylation, we just attach a preformed oligosaccharide on the amide nitrogen of asparagine. So basically, this is N-glycosylation and O-glycosylation. Now, one function of O-glycosylation is it is involved in protein to protein interaction or signaling. So we can say it's involved in cell to cell signaling. That's one function of the O-glycosylation. So, and it's also involved in cell adhesion and signaling. It's involved in lubrication and protection of the epithelial cells. So those are some of the functions of O-glycosylation. All right, so we are going to look at the differences between O-glycosylation and N-glycosylation. All right, let's look at the differences between N-glycosidic linkages and O-glycosidic linkages. Now, one, asparagine is the linkage site. Remember that in N-glycosidic linkage, the sugar, the oligosaccharide, is linked to the amide nitrogen of asparagine, while in O-glycosidic linkage, the sugar is linked to the hydroxide group of either serine or threonine. So serine or threonine is the linkage site. Then here, the first sugar in the oligosaccharide linked is N-acetylglucosamine, while here N-acetylgalactosamine is the first sugar attached. Now here consensus sequence is asparagine X serine or threonine. Now it means the sequence of attachment must be in this format: asparagine, then any amino acid other than proline, then serine will come next, or threonine. So it must be asparagine, then one amino acid before serine or threonine. It must be like this. But in the O-glycosylation, there is no consensus required. There's no preformed step required, or this is what must come before this. Nothing like that in O-glycosylation. Number 4, location of initiation is endoplasmic reticulum. Remember that N-glycosylation starts in endoplasmic reticulum and continues in Golgi apparatus. But in O-glycosylation, the location of initiation is the Golgi apparatus. So it's the Golgi apparatus. And this one requires extensive trimming and modeling. Remember, what we are attaching is a preformed oligosaccharide. So we have to trim and model the oligosaccharide that we want to attach. But here in O linkage, we are attaching the monosaccharides one after the other. We are not attaching a preformed oligosaccharide. We are attaching the sugars one by one. Then this one requires a lipid carrier, while O-glycosylation does not require a lipid carrier. So these are the differences between N-glycosylation and O-glycosylation.